So the function of the immune system is all about immunocompetence. This is where the lymphocytes understand exactly what they're supposed to do, what antigen markers they've come in contact with that are pathogenic, and which antigen markers they've come in contact with which are self. When we look at immunocompetence, it's all about the lymphocytes, and what to remember is that a majority of lymphocytes will be T cells, whereas a minority of lymphocytes will be B cells. The T cells will mature within the thymus, whereas the B cells will mature within the bone marrow in terms of differentiation and specialization. As we age, the thymus will atrophy away. This atrophication is going to minimize the amount of new T cells that can be derived as we age through life. So let's take a look at the maturation of the system. The immune system itself is constantly maturing. It's the last system to fully mature. Early development occurs through exchange of IgGs from the mother or from the mother to be through placental exchange. This is where we can have mismatch issues with Rh factors. The other thing that will start taking place is that the fetal immune system will start to learn self-antigens, as is, this is what cell belongs to me. Following that, we then get the first stages of passive immune acquisition, passive immunocompetency. And this occurs through exchange of antibodies from the mother through breast milk, followed by vaccination sequences that start to take place around 6 months of age. In terms of full maturation, we actually don't get a fully mature system until death, as the system is constantly maturing based off of constant interaction with pathogens that it gets exposed to. So let's take a look at the ability to self-recognize and self-tolerate, which is the key feature of immunocompetence. So we start off with the immature T cell. The immature T cell, we want them to be able to recognize self, but not bind and become active to self. So we need it to recognize self because the T cells itself will undergo an attack on any antigen that it does not recognize. And so we need it to activate on the unrecognizable antigen, but we need it to not activate on the recognized antigen. Any T cell that does not follow this sequence of events, meaning that it will start to activate upon exposure to the self antigen, will attract macrophages into the region where it's undergoing its maturation in the thymus and will be destroyed, hopefully. Those that undergo the full maturation will then become active and get released into circulation. Once the mature T cell undergoes a recognition of a non-self antigen, it undergoes a stimulation. This stimulation leads the mature T cell to becoming an activated T cell. Any T cell that doesn't follow this sequence of events will undergo an apoptotic sequence of events because it's not being used. The activated T cell will undergo a proliferation and differentiation into two forms, either the helper form or the killer form. While most will become activated upon stimulation, some will undergo what's referred to as a sequestration. The sequestration will regulate T cell activation and will form the memory T cells for subsequent exposure to that same antigen. Any helper or killer T cell that is around following the activation sequence will go into a dormancy phase and will become the memory T cell. Sometimes the sequestered T cells will secrete hormones that will actually truncate or regulate the T cell activation throughout the body so as to make sure that we have the correct level of stimulation taking place. The other form of activations with the B cells. The B cells are slightly different than the T cells. With the B cells, we don't want them to be able to recognize self, because if they recognize the antigen for response, they will release an antibody in response to that recognition. So anything that within the B cell that will activate upon exposure to the self antigen will undergo the apoptotic cycle through macrophagic activity, because we need it to not recognize self so that it will activate upon recognition of non-self. Those B cells that undergo maturation will get released into circulation, and upon exposure to the first antigen marker it activates to, it will undergo stimulation. Just like with the T cells, the stimulation leads to an activated B cell. This activated B cell will undergo proliferation into its clonal as well as its plasmal forms, releasing immunoglobulins as well as distinct hormones for secondary complementary activation. Just like with the T cells, we'll see some that get sequestered. These sequestered B cells will undergo a regulation signal that basically holds them within the lymphatic tissues, in particular within the lymph nodes, until secondary signaling comes into play upon next exposure to that same antigen. The B cell activation in this case here, in regards to the immunoglobulin, the assemblage of the immunoglobulin will be specific to the antigen that was activated in terms of the sequence of events causing the B cell to release the immunoglobulin. Every B cell that has been exposed to that antigen will be able to create an antibody or immunoglobulin that is specific to that antigen. Many B cells will be able to produce immunoglobulin markers and antibodies to multiple types of antigens through having a variety of genetic and epigenetic regulations in the protein synthesis process, forming the antibodies.